I'd like to introduce Dr. Yong Xu. Dr. Xu is an associate professor of pediatrics and molecular and cellular biology. His lab investigates how neurons in the hypothalamus regulate appetite and satiety in mammals, including the identification of drugs and small molecules that can regulate hunger and appetite. This work is vital to strategies to combat current important public health concerns, such as obesity, diabetes, and eating disorders. Congratulations to Dr. Xu. I invite you to learn more in this video presentation on his recently published work.
I want to start by saying that I feel extremely honored to be able to stand here to receive this DeBakey Award and also have this opportunity to share a little bit of our research. So my lab is being interested in a very broad question, that is how the neurons in the brain control the whole body metabolism. And this is highly relevant to a number of human medical conditions, such as obesity, including obesity-associated metabolic problems, such as diabetes. Usually I will use a different slide to make a justification why obesity is such a big problem. For example, I could say CDC recently reported that about 42% of the US adults are now considered to be obese. That is a really alarming rate. But given we're in the middle of COVID-19 crisis, I decided to use this slide here, where they are actually our neighbor state, Louisiana, did a study to look at mortality rate of COVID-19 patients in their state. And as we often heard these days from television or conference, is that the people who unfortunately pass because of the COVID-19 are those people who have certain underlying conditions. But probably what people have not really appreciated is the top two risk factors associated with COVID-19 mortality are diabetes and obesity. So obesity is not really just a cosmetic issue. In crises like this, obesity is actually a risk factor that determines you live or die. So our group is interested to understand why certain people develop obesity and whether we can develop a cure for this disease. So when we think about human obesity, there's a fact that is often underappreciated. That is, for most of the human obesity, is actually a brain disease. This is a Nature article published in 2015, where they summarize all the GWAS study showing association between the human genomic variance with human BMI, the body mass index. What is striking as shown here is that 95% of genetic variants associated with human BMI actually affect genes that are highly expressed in the nervous system. In other words, for most of humans who have obesity, they have something wrong in the brain. And so among all this genetic variants that has been associated with human obesity, actually so far only a few of them has been definitively tested to prove they are the cause of human obesity. But within this few identified genetic basis for human obesity, many of these genes actually affect this group of neurons that I'm going to talk about today. And their name is melanocortin neurons. So what are the melanocortin neurons? So seen here is a coronal section of a mouse brain. It's coronal section doesn't mean the coronal virus. And so this is just a way that we cut the mouse brain. So what you're seeing here is a basal medial hypothalamus, a mouse hypothalamus. And around this third ventricle, there's this triangle structure. This is called arcuate nucleus, or ARH. So within this region, you will see this red neurons. These are the pre-opium melanocortin neurons, or POMC neurons. And adjacent are these green neurons. These are the neurons that release this neuropeptide called a Goody-related peptide, or we call them AGRP neurons. Both AGRP neurons and the POMC neurons are melanocortin neurons. And this is a very simple schematic demonstrating how we currently understand about the functions of AGRP and POMC neurons in the regulation of our feeding behavior as well as the body weight balance. Typically, when we are well fed, these POMC neurons, they will start to fire more action potentials. This will engage a release of their neurotransmitter. One of them is called alpha MSH. Alpha MSH is actually one of the gene product of the POMC gene. By the way, the POMC neurons are called POMC neurons because they express POMC gene. And one of the gene product is alpha MSH, which is a neurotransmitter or neuropeptide. So alpha MSH will act on their downstream neurons that express its receptor called the melanocortin 4 receptor, or MC4R. So this will lead to the activation of these downstream neurons, which ultimately leads to a satiety signal. So this, in a large degree, explains when we are well-fed, we stop eating. On the other hand, when we feel hungry, these adjacent AGRP neurons will start firing more action potentials. They will release one of their neurotransmitters called GABA, which is an inhibitory neurotransmitter. And this GABA will act on the adjacent POMC neurons to inhibit the POMC neuron firing activity. At the same time, AGRP neurons will also release this neuropeptide called AGRP. As it turns out, AGRP is an endogenous antagonist of the same receptor, MC4 receptor. So AGRP will inhibit neurons that express MC4 receptor, and they will lead to the appetite signal. So this will explain when we are hungry, we want to eat. So as you can see, this little yin and yang system, AGRP and POMC neurons in the arcuate nucleus, they dynamically respond to our nutritional status every day, and then they control our feeding behavior in a proper fashion in order to keep a balanced energy homeostasis. And our work since 2008 has been pretty much focused on the intracellular mechanisms or external signals that can coordinate the function or firing activity of these melanocortin neurons in all kinds of conditions to try to keep the balance of energy homeostasis. So today I'm going to share you two little stories that we recently published, which both focus on the POMC neurons. First, I want to show you two literature just to make a case of why POMC neurons are so important. So this is a work published by a Chinese group about 8 years ago or 7 years ago. What they did is they genetically killed all POMC neurons in these animals, as you can see here. So if the animals don't have POMC neurons, they will eat about 5 to 6 grams of regular chow diet every day. If you don't know how animals or mice usually eat, this is the control group down there. All these control groups, they eat about 3 grams regular chow every day. So now you can appreciate, for animals that don't have POMC neurons, they eat extremely, actually double the amount of food every day. That's a lot of food for them to eat every day. If they keep doing that for a certain period of time, obviously they're going to develop massive obesity as demonstrated here. So this data really indicates that the POMC neurons are absolutely essential to prevent overeating, to prevent obesity. And we know that the POMC neurons are called POMC neurons because they express POMC gene. This is actually one of the fundamental functions of these neurons. So then the question is whether POMC gene expression is also important for body weight control. The answer is yes. And this is a study published by a Michigan group about 7 or 8 years ago, where they genetically delete POMC gene expression only in the arcuate nucleus, as you see here. And this is a control animals that shows normal POMC gene expression in the arcuate nucleus. So if the animals lose POMC gene expression in the arcuate nucleus, again, they develop this massive obesity. Actually, the same thing holds true if this is a human who have a loss of function of POMC gene expression. They have diminished POMC gene expression. They're going to be massively obese. But of course, there are many factors that regulate POMC gene expression. And then loss of function of these regulators and modulators may not diminish the POMC gene expression, but may substantially reduce POMC gene expression. And that will also lead to a certain degree of obesity. And that is exactly what we show in one of our recent studies. In this study, we collaborated with Dr. Salah Faruqi from the University of Cambridge. Salah was able to identify a number of missense mutations in this gene that we've been working on called steroid receptor coactivator 1, or SRC1. So this is a schematic demonstration of the protein motif of SRC1 protein, and then you can see these mutations cause the amino acid changes in this protein, and this is all identified from obese children. And one of the things we did is to make a mouse model which mimic one of these mutations identified from obese children, which is this one here. And then we did this with the help from Dr. Yong Xu at Molecular Cell Biology, and then we did this through Cas9 CRISPR-Cas9 technology. So what we found is if these animals carry this missense mutation that is observed from obese human, these animals also become obese, as demonstrated here. So in my opinion, this is perhaps the most definitive evidence that we can come up with to prove the notion that this mutation identified from human really is the cause to lead to the obesity in at least this subset of patients. And then we went on to try to identify what is the underlying mechanism. And then we start to realize that SSC1 actually normally functions as a modulator of POMC gene expression. It's required to stimulate POMC gene expression. And the majority of these mutations identified from human obese children, they actually damage this function of SSC1, and therefore they need to reduce POMC gene expression, as demonstrated by this POMC luciferase activity assay shown here. And because of this, we then made an animal where the SSC1 is specifically knocked out only from POMC neurons, nowhere else. And by doing this, we found the animals also become obese. This exactly recapitulates what is seen in those patients who carry the SSC1 loss function mutation. So this example really demonstrated that the expression of POMC gene is so critically regulated, and the mutations in modulators of POMC gene expression could lead to human obesity. And as we're talking about the fundamental function of POMC neurons, of course, one of them is to express POMC gene, but the other one is to firing action potential. Actually, for that matter, firing action potential is one of the fundamental functions for pretty much all neurons, because only by firing action potentials can these neurons release their neurotransmitter or neuropeptide to talk to their downstream neurons. So for that reason, we're also very interested in looking at the firing activities of POMC neurons. And by doing those, and we start to realize that there is a very interesting sexual dimorphism in terms of POMC neuron firing. Shown here is a typical firing activity of a male POMC neurons, and this side is the female POMC neurons. So you can obviously appreciate that the female POMC neurons firing a lot more than the male POMC neurons. And this is a quantification from hundreds of neurons that we recorded which make that point. So this got us interested in looking into these POMC neurons and to figure out what is the molecular mechanism that are underlying this sexual dimorphism. And we did a number of genomic screening as well as validation approach, which eventually led us to focus on this pair of transcripts. One is called TAP63, which is a lot higher in female POMC neurons than male POMC neurons. The other one is delta N P63, which is a lot higher in male POMC neurons than the female POMC neurons. So what are they? So both these transcripts actually are promoter variants of the same gene called P63, which encode a transcription factor. As it turns out, TAP63 is an active form of this transcription factor. Basically, they transcribe genes. And delta N actually is a dominant negative form, which they inhibit the activity of TAP63, transcriptional activity. So by putting this together, this pattern basically indicates that in female POMC neurons, there is a lot higher transcriptional activity of this transcription factor, TAP63. And then we start to wonder whether this sexual dimorphic pattern in terms of TAP63 transcription activity may explain the sex differences we observe in terms of POMC neuron firing. To test this, we generated animals where the TAP63 is specifically knocked out from POMC neurons. And then we start to characterize their phenotypes. Shown here just one key phenotype we found from this animal, which is the body weight. Shown here is the male animals, we have control animals, we also have the knockout animals where TAP63 is only knocked out in POMC neurons. So we fed these animals on regular chow diet from weaning all the way till the age of 30 weeks, and then we switch it to high-fat diet feeding. As you can expect, that the high-fat diet feeding dramatically increased the animal's body weight. This is a very typical phenomenon in our field, and we call this diet-induced obesity. Actually, people commonly use this paradigm to model the human obesity, because most of the human obesity are characterized as increased feeding of high palatable food. So we see what we typically see. That is, the high-fat diet feeding can induce obesity development in these male animals. However, what is boring about this data is there is no difference whatsoever between our control and knockout males. Then when we look at the female animals, the data become more interesting. And when the animals are fed on regular chow, we still see no difference between our control and the knockout females. However, when we switch them to high-fat diet, we notice that the female knockout mice start to gain a lot more weight compared to the female controls. So this indicates that the loss of TAP63 only in POMC neurons can increase the sensitivity of these female animals to develop obesity when they were fed with high-fat diet. But the same phenomenon is not seen in male animals. Now, I'm showing you exactly the same data, but in a slightly different way. Here, we're calculating how much body weight gain the animal had during this period of high-fat diet feeding. Here is the control animals, the male versus the control female leader mates. So control male animals can gain 20 grams of body weight during that short period of high-fat diet feeding. That is a lot of body weight to gain. But what is striking is their control female leader mates gain about 10 grams, 50% of the male body weight gain. This again is a very typical phenomenon that we see in this field. That is, the female animals are more protected from diet-induced obesity, but nobody really knows why. And then when we look at the knockout animals, this is very striking. By knocking out the TAP63 only in POMC neurons, we found regardless if this is a male or female animals, they gain about 20 grams of body weight during that same period. What that says is that the loss of TAP63 in POMC neurons can diminish the sex differences in terms of body weight gain in response to high-fat diet feeding. And then we went on to look at the mechanisms of this phenomenon, and eventually we sat down to this model. We believe at the molecular level, the POMC neurons, they are sexually dimorphic in terms of the level of TAP63 and the delta N. And specifically, the female POMC neurons has high TAP63, and the male POMC neurons has high delta N P63. And then this molecular sexual dimorphism actually leads to a sexual differences in two fundamental functions of these POMC neurons at the cellular level. This includes expression of POMC gene expression, which I didn't show you data today. And the other thing is the firing activity of these POMC neurons. So both this fundamental function of POMC neurons are enhanced in female POMC neurons, and this is both due to the increased TAP63 transcription activity in female POMC neurons. And then this cellular function, enhanced the cellular function in female animals, ultimately translated at the physiological level that these female animals are more protected from diet-induced obesity compared to the male counterparts. All right, so finally I want to just summarize what I shared with you today, just to make a very general point. One is we believe that human obesity largely originated from brain dysfunctions, but a lot more needs to be studied and learned from this area. And also we want to emphasize that sex matters in neural control of the metabolism. Actually, in that matter, I think sex matters for pretty much most of the physiological process that we look at. We just have to look at them carefully and properly. And finally, more specific to the melanocortin neurons that are my favorite population, and through the work that I presented today, also our previous work, we're convinced that the melanocortin neurons, they're very fragile in terms of all kinds of stress, including overnutrition or genetic variants. Therefore, the dysfunction of the melanocortin neurons often underlie the development of obesity. And for the very same reason, we think that these melanocortin neurons are very attractive drug target for the obesity treatment. Okay, finally, this is perhaps the most important slides that I have to spend most time on, and then I can't get here without help from the people I've been interacted over the last more than 10 years. I want to start it from my mentors. I really have to thank most of the mentors. I have many of them, and particularly I want to thank my postdoc mentor, Dr. Joe Almquist from UT Southwestern at Dallas, who brought me in this field and taught me how to do science in this field. And also, I want to thank the mentors that I have in Baylor community. This includes my CNRC director, Dr. Dennis Beer, as well as Dr. Bert O'Malley and Dr. Mingzhe Cai. They're not only my leaders, but also my collaborators in many cases. And more importantly, they're my role models. They taught me how to collaborate with others, how to lead my lab, how to lead in my community or in my scientific field. And of course, I need to thank the people who have helped us all along the way of all this project. Here I just highlight the people who are relevant for this melanocortin projects that we've been working on, but there's many other people which are not on this list because we're not talking about the relevant research project today. And this includes a lot of people within Baylor, as well as people in Texas Medical Center or all over the world. And finally, I have to say I was very, very fortunate that I have the opportunity to work with a lot of young, bright, and diligent people in my group, and really it's their efforts that leads to the little bit of advances that we were able to make in our field. And these are their names here. And finally, I want to thank all the funding sources that have been so generous so far to support our research.